Hello guys, welcome to Dr. Science. So today we are going to talk about the metabolism of acylglycerols and sphingolipids, and this is taken from your Harper's Biochemistry textbook, and the chapter is chapter 24. And in this video, which is the first video of this series, we are going to talk about the overview of acylglycerol biosynthesis. So please watch my video in a chronological order, or else you will not understand a single thing here. Okay, so this is the diagram that is given in your biochemistry textbook. So as you can see, this is the overview of acylglycerol biosynthesis. So I will try to simplify this diagram as much as possible, so don't worry about it. But first you have to understand the structure of the cell membrane. Okay, now we are going to see the structure of the cell membrane. Usually our cell membrane is made up of two layers. Okay, so here is a phosphate, and this phosphate is usually hydrophilic. By hydrophilic I mean it means it loves water. And on the other hand, it have lipid. Usually the lipid is lipids such as your fatty acids are present. Okay, now this fatty acids or the lipid component is hydrophobic. It means it hates water. Okay, so how is the cell membrane arranged? So usually our cell membrane, the cell membrane is made up of lipid bilayer. Okay, it means it have outer leaflet, and not only that, it have an inner leaflet. Hence it is called as the cell membrane is a lipid bilayer. Okay, so as you can see here, the outer leaflet have phospholipids. Okay, and usually, so this is the phosphate and this is the lipid. I am going to draw with a different color here. This is the lipid component. And similarly, the inner leaflet will also contain a phosphate and a lipid, and here is another phosphate, and here is your lipid. Okay, so in the outer region or the outer leaflet, there are mainly phospholipids such as your sphingomyelin, and the other one is your phosphatidate choline. Okay, so in the outer leaflet we have the sphingomyelin and the phosphatidate choline, whereas in the inner leaflet we have phosphatidate serine, and we have phosphatidate ethanolamine. Okay, once again I will summarize once again. So in the outer leaflet and the inner leaflet, outer leaflet were consisting of phosphatidate choline and sphingomyelin. Okay, and the inner leaflet were consisting of phosphatidate serine and phosphatidyl ethanolamine. Okay, so these are your phospholipids. Now, where these phospholipids are coming from, we have to understand the synthesis of these phosphoglycerols or the phospholipids. Okay, in order to understand this, we have to go a little bit deeper, because this is a very tough topic to understand. So we will start from glycolysis. Okay, as you know, from glycolysis we learned that from a single glucose molecule we are going to synthesize glycerol 3-phosphate and dihydroacetone phosphate, or simply DHAP. Okay, and this glycerol 3-phosphate and dihydroacetone phosphate, they are interchangeable. It means dihydroacetone phosphate can be converted into glycerol 3-phosphate, and dihydroacetone phosphate can be converted into glycerol 3-phosphate, and similarly glycerol 3-phosphate can be converted into dihydroacetone phosphate. Now this glycerol 3-phosphate is also known as your triose phosphate. From this glycerol 3-phosphate or the triose phosphate, if we are going to do acylation, a process known as your acylation. So in simple terms, if we are going to acylate triose phosphates, then we can synthesize compounds such as your triacylglycerols or your phosphoglycerols, or we can as I already told you, the phospholipid content such as your phosphatidyl choline, phosphatidyl ethanolamine, sphingolipids, and your phosphatidyl serine. Okay, so from glycerol 3-phosphate or the triose phosphate, if we are going to do acylation of the glycerol 3-phosphate, we can synthesize the phospholipid bilayer. Okay, this is what that is given in your biochemistry textbook. Now we have to understand how the acylation process is done, and how from glycerol 3-phosphate we can synthesize the triacylglycerols and phosphoglycerols. Now here I want to also mention that usually this is a glycerol, and to this glycerol there are 1 2 3 there are 3 fatty acids. Now this entire component is your triacylglycerol. Okay, now if triacylglycerol will undergo hydrolysis, okay, there is an enzyme which will cut this triacylglycerol, and the name of this enzyme is a lipase. So lipase will do hydrolysis of the triacylglycerol, and whenever this happens, whenever the lipase is going to cut the triacylglycerol, it will usually produce 2 components. As I already told you, one of the component was glycerol, and the other component was your fatty acid. Eventually this fatty acid will go into the circulation and it will bind with the So this was the fatty acid, and eventually this fatty acid will go into the circulation and it will bind to the albumin and it will reach to the target tissues. Now as you can see here, we have this glycerol component, and if this glycerol by an enzyme called as your glycerol kinase, okay, this enzyme is very important, because glycerol kinase activity is absent in some of the tissues, and those tissues which have the deficiency of glycerol kinase enzyme activity, they cannot convert the glycerol kinase into glycerol. Actually, I want to say one more thing here. This glycerol here is a 3-carbon unit. It means it have 3 carbons present on it. So glycerol kinase will put a phosphate on the 3rd carbon. So once again, here 1, 2, 3, the 3-carbon unit, which was the glycerol, right? And glycerol kinase, this is the glycerol kinase, and glycerol kinase will put a phosphate on the glycerol. So here on the 3rd carbon it will put a phosphate. So this newly synthesized molecule is your glycerol 3-phosphate, because on the 3rd carbon it will put a phosphate. Okay, so glycerol is converted into glycerol 3-phosphate by glycerol kinase. Now eventually this glycerol 3-phosphate will get converted into a compound known as your phosphatidate. Okay, this is very important. Why this is important? Because this phosphatidate is a common precursor for everything. It is a common precursor for everything. By everything I mean, from this phosphatidate we can synthesize diacylglycerol, and from this diacylglycerol we can synthesize triacylglycerol, and similarly we can synthesize phosphatidyl coli and phosphatidyl ethanolamine, which were the components of your lipid bilayer. And not only that, from this phosphatidate we can also synthesize a compound called as your cardiolipin. Now why this is important? Cardiolipin is usually present in the inner leaflet of your mitochondria. Okay, so if I know you will hear the cardiolipin in your internal medicine, and usually there you will hear the word cardiolipin very frequently, which is used as a marker for diseases. Okay, so this is usually present in the inner leaflet of mitochondria. Okay, cardiolipin is also a biomarker for apoptosis. And from this phosphatidate itself we can synthesize other components such as your phosphatidyl ionocytol. Okay, so from phosphatidate we can synthesize diacylglycerol, triacylglycerol, phosphatidyl choline, phosphatidyl ethanolamine, and not only that, cardiolipin, which is present in the inner leaflet of mitochondria, and the phosphatidyl ionocytol. So we can say that phosphatidate is a very common precursor for everything. So eventually, once again, from glycerol we can synthesize glycerol 3-phosphate by the enzyme glycerol kinase. Okay, and from glycerol 3 phosphate we can synthesize phosphatidate, and from phosphatidate is the common precursor for everything, because it will synthesize the diacylglycerols into triacylglycerol, and from diacylglycerol also we can synthesize the phosphatidylcholine or phosphatidyl ethanolamine. And from phosphatidate we can synthesize the cardiolipin, which is present in the mitochondria. And finally we can synthesize phosphatidyl ionocytol. Okay, now the main question is, I already told you, in some of the tissues such as your muscles and adipose, this glucose kinase enzyme is deficient or low in number. So in these muscles or adipose tissue, how can we convert the glucose the glycerol into glycerol 3 phosphate, and we can synthesize the acylglycerols. Okay, now in order to understand this, I already told you the glycolysis pathway, where glucose was getting converted into two components. One was your glycerol 3 phosphate and the other was your dihydroacetone phosphate. As I already told you, glycerol 3 phosphate can be converted into dihydroacetone phosphate, and similarly dihydroacetone phosphate can get converted into glycerol 3 phosphate. Okay, so in muscles or adipose which lack the enzyme glucose 3 kinase will have a special enzyme called as your glycerol 3 phosphate dehydrogenase. Okay, this enzyme, what is the function of this glycerol 3 phosphate dehydrogenase enzyme? So as you can see here, this is glycerol 3 phosphate and this is the dihydroacetone phosphate. This dihydroacetone phosphate will get converted into glycerol 3 phosphate and during this step ATP is utilized. Okay, so here NAD is will get converted into NAD plus, and the enzyme here responsible is your glycerol 3 phosphate dehydrogenase. Okay, so eventually from this glycerol 3 phosphate we can synthesize your phosphatidate, and from phosphatidate we can synthesize all your acylglycerols, and acylglycerols such as your triacylglycerol, diacylglycerol, etc., and your phospholipids such as your phosphatidylcholine and phosphatidylethanolamine. Okay, I will simply put this one as phosphatidate here. Okay, so once again, in the muscles we lack the enzyme glucose 3 kinase, hence we have the other enzymes such as your glycerol 3 phosphate dehydrogenase. Okay, I hope you are clear with this. So this was a brief introduction on the overview of acylglycerol synthesis, and in my next video I'll dig more deep into the chapter, but don't worry. And here here the enzyme is glycerol 3 kinase. Okay, don't write glucose 3 kinase because I was a little bit confused regarding the glucose 3 kinase and glycerol 3 kinase. So in my video, if I told glucose 3 kinase, please write it as glycerol 3 kinase. Okay, and here I will just show you this diagram here. So as you can see here, glycerol 3 phosphate, which is a triose phosphate, can be by acylation can get converted into phosphatidate, which is the common precursor for acylglycerol synthesis. And what are the acylglycerols? Acylglycerols such as your diacylglycerol, from which we can synthesize the triacylglycerol. And not only that, we can synthesize phospholipids such as your phosphatidylcholine, phosphatidylethanolamine. And not only that, from phosphatidate we can synthesize your cardiolipin. Oh my god, we can synthesize the cardiolipin. And from phosphatidate itself we can synthesize phosphatidyl inositol. As you can see here, this glycerol 3 phosphate, so glycerol 3 phosphate and dihydroacetone phosphate, if muscle lacks the enzyme glycerol 3 kinase, the dihydroacetone phosphate can get converted into glycerol 3 phosphate by glycerol 3 phosphate dehydrogenase. Okay, I hope I am clear with this chapter, and in my next video, which is going to be a very long video, I will discuss about the biosynthesis of triacylglycerols and phospholipids. Thank you guys.